Hello everyone, present here virtually. I am Rutuja here and I welcome you all for today's session, which is titled as Designing PCR Primer Using Bioinformatic Tools. And presenting this session, we have with us our today's expert, Dr. Sandeep Debinath. And present Dr. Sandeep Debinath is experienced assistant professor at the Department of Genetics and Plant Breeding, Palli Shiksha Bhavan Institute of Agriculture, Vishwa Bharati University, Sri Niketan, West Bengal 731236, India, with extensive expertise in higher education sector. He has expertise in both plant breeding and genetics. Strong education professional having a PhD in genetics from Vidhan Chandra Krishi Vishwavidyalaya. Additionally, Dr. Debnath is active in number of publications and conference presentations, and he has already authored 30 research articles, 16 patents, 2 books, and 6 books chapters. His area of study include bioinformatics, mutant breeding, biotechnology, and AI-enabled plant breeding. He has supervised 2 research projects. Sir, you can start now.
Okay, so thank you everyone. Hello friends. Very good morning to all of you. So today I'm going to deliver my lecture on designing PCR primers using bioinformatics tools. I am Dr. Sandeep Debnath, so welcome you all. So designing PCR primers is a kind of routine work to all the molecular biologists and many researchers. Okay, those also in the field of medicine, in the field of genomics, in the field of microbiology, in the field of plant breeding, and so on, so on. So actually, today I will show you the most powerful bioinformatics tools to design PCR primers. Okay. So, just for confirmation, I have just changed the slide. So, is it changing? Hello, everyone. Is it changing?
Yes, sir.
But I have changed it. This is the problem. So, is it changing? Or the same slide?
Same slide, sir.
Same slide. So, I think. Now?
Yes, sir. Change.
Okay, so can you see the brief history, this slide?
Yes, sir.
Okay. So, actually, before going to PCR primers, I need to discuss about the PCR. PCR, you all know that it is polymerase chain reaction. I will come later that what is this chain reaction and everything, this all these things. But before going to that, actually PCR is a game of controlling temperatures. You have a set of reacting agents. Okay, so actually you have to go for some chemical reaction. Okay, that means DNA replication. But in a PCR machine is a actually it will control different temperature in a very short period of time. That's why this is the beauty of the machine PCR. But the DNA replication also occurs in natural cellular condition also. But there, actually, we live in such an environment where DNA replication is going on. Our environment, what is the temperature? Maybe in summer, maximum 40-45 degrees centigrade. Okay, or maybe in winter it is maybe 3 degree, 4 degree, 5 degree. I am saying in general. Okay, so this is almost the habitat condition, the natural condition where we live. We live, our plants live, our crops live. So our body system, our cellular system, they are also habituated with this. Okay, so with this temperature regime. Okay, with this temperature range. So all the proteins, all the enzymes that work in our cellular system, they are also habited to work with these temperature range. But in PCR, I have already told this is a game of controlling temperature. In PCR, we need such an enzyme, such a polymerase that is able to, I will rather say, survive. That means it will not degrade or degenerate, whatever you say. That will function in a very high degree of temperature, that is about 94 or 95 degrees centigrade. Because for denaturation step, you need to follow this temperature. Okay, so that's why people searched that what kind of polymerase we can use. If the denaturation temperature, suppose it was not like that high, then we could have performed this PCR reaction with our normal polymerases that is available or that is found in our body system, maybe in our known microbes or from the plants. Okay, whatever we see around us. But here, as we have to put a temperature of 95 degree, 94 degrees centigrade, we need such a polymerase. That's why from hot spring, people isolated these stack polymerase and then thought, okay, it can be used. It can be used in the PCR machine where actually I am controlling the temperature. Okay, from high to high, medium, and that is 95 degree, suppose 55 degree, then 72. Again 95, 55, 72, like this. So it is very high temperature job. So people successfully could isolate these stack DNA polymerase, very globally known as stack DNA polymerase, okay, from bacteria which is grown in hot spring. Okay, so this is the first thermostable enzyme. Then people have done lots of mutation to that sequence, and they have made different categories of this stack polymerase. Okay, that is by doing site-directed mutagenesis, by doing some different mutations, they have changed some protein structures, and with different modification, people now could able to artificially evolve such thermostable enzyme, which are where the fidelity is very high, where the efficiency of doing polymerization is very high. Okay, that has been achieved till date. So it was commercialized, this stack DNA polymerase. Okay, and during 1989, it was regarded as the molecule of the year. Okay, because of its high demand. Okay, until now, people also there are many multinational companies, though, or biotech companies, those who manufacture this stack polymerase, they are still in the process of doing different mutations at different sites to make this thermostable enzyme, that is stack polymerase, to make it more and more efficient. And according to their efficiency, the price depends. When being a molecular biologist, when you will go for buying this enzyme for your laboratory, you will find there is a different grades of stack polymerase are available in the market with different price. So depending on their efficiency for doing its job of polymerization, the price varies. Okay, so I think it's clear to you. So these actually this discovery has made a revolution in this biotech industry. Okay, so I must take the name of Kagi Mullis, who was awarded with the Nobel Prize. So before going to primer designing, let's just recall our knowledge about PCR. So what actually we do during PCR? Okay, we have our DNA of interest. We have template DNA. Suppose I have taken one, maybe one human genome, maybe mouse genome, maybe for different, suppose rice genome. I have extracted the whole genome. I have extracted by DNA extraction method. Suppose. So this is the initial stage one molecular biologist do. So from that, genome is very huge, very big in its own length. So suppose I want to, there are millions of bases in a genome. But my target is, suppose I want to just amplify one SSR, which is just 200 base pair long, or maybe anything just 500 base pair long. So how to do that? Besides the PCR primer, almost all other things, they are almost unique for each and every PCR reaction. That means you have to add the buffer, you have to add the DNTPs, all other ingredients. Okay, you have to add the water. Okay, you have to add the template. Almost this is unique. Okay, maybe in some specific cases you have to add some magnesium ions. Okay, additionally for more specificity, whatever maybe. But the main success, the key thing for the success of a PCR reaction is the PCR primer. That means that researcher, that scientist, that molecular biologist who is able to design the PCR primers, the forward and reverse primers, the most efficiently, actually he will get the success. Assuming that he has done all other experiments very carefully, okay, there is no error. But actually, the bands, actually the success of amplification, it will principally depend on how good your PCR primer, your forward and reverse primer is. So actually, you have to first, you have to make the two strands of DNA separated. Okay, that is done just by the game of controlling temperature. That means if you put your genome or your DNA of interest, that means in a temperature range of 94 or 95 degrees centigrade, it will denature. That means the two strands which are joined by hydrogen bonds, they will just separate. Okay, now they will act like single stranded. That means the complementary bases, they are not joined now. They have been separated. And now you will put the primers. Here you can see, you will put the forward and reverse primers. They will go and bind. This is kind of theory. Okay, so after the first cycle, that means just after annealing of primers, if you again just elevate the temperature to 72 degrees centigrade, the polymerization will start. That means one by one, just seeing the complementary strand, the DNTPs will, addition of DNTPs will go on like this. Okay, so like this, by this forward and reverse primers. So after 20 to 30 cycles, you will get the millions of copies of your desired sequence. So this is very easy. So here you will maintain for denaturation 95 degrees centigrade for denaturation. Then for annealing or addition of your primers to that template, that will be done at in a range of 55 to 60 degrees centigrade. And then if you again elevate the temperature to 72 degrees centigrade, the reaction will go on. And in a repeated cycles, actually the PCR reaction will happen. And if you load this thing in a jelly electrophoresis, you will be able to view the bands. So this is very simple. Okay, so this is actually the steps of PCR that I have already discussed. Okay, that means denature. Don't forget these things. Denaturation at 95, then primer annealing at 55, and then the synthesis of new strands or the extension, whatever you say. Okay, so it will go on after binding the polymerase. Then the PCR reaction will go on. But now my question is that what if, suppose the length of this forward primer, maybe this is the reverse primer, they are not identical. Maybe this is 18 BP length, maybe this is 22. So obviously, the annealing temperature, suppose for this forward primer, it is 56 degrees centigrade, and somehow the annealing temperature of the reverse primer, suppose it is 62 degrees centigrade. That means the temperatures are different of annealing. But during PCR, during this step, you will be able to put only a single temperature. Okay? So for one primer, if the annealing temperature is 56 and for another if it is 62, then actually what temperature will you set at this step? This will create problem for your PCR reaction, and it happens also. Whenever you do, whenever you make the PCR primers manually, this thing will happen. Okay, you cannot every time you cannot put the temperature, because the actually the temperature of annealing temperature depends on, there is a thumb rule, I think you know, that is 2 into A plus T plus 4 into G plus C. That means how much A and T is present, you multiply it with 2 plus how much G and C is present in the primer, you multiply it with 4. Okay, so ultimately this is the annealing temperature. So in your forward primer, suppose the AT content is more, and your reverse primer, suppose the GC content is more. So obviously GC is multiplied by 4. So that means the annealing temperature of this primer, it will be more. So you definitely, it will create problem when you will either you have to set 62 degree here or you have to set 56 degree here. So these temperatures are not synchronized, it will create problem. So there are lots of issues that you cannot handle at a time manually. You have to take the help of different tools. Tools means bioinformatics tools. Another problem, I am just think of it. We'll answer everything later. Suppose you have designed one primer, suppose this is forward and this is reverse primer, and your intention, your anticipation is that it will go and bind here on. But genome is written, genome only by A, T, G, and C. Only 4 options are there. So in that huge, this long genome, the length is very huge of a single genome, you will find similar regions in many where in that genome. That means, so you are thinking, I have designed primers from this region, so it will go and bind here only. No. This primer may have sequence complementarity with, suppose, with another 10 regions of your genome. So whenever you will control the temperature to its annealing temperature, the primer will not go and bind to your target region only. It will go and bind another 10 regions where there are complementarity. So instead of getting a single band as per your expectation, you will get actually 11 bands. So how to identify what is your target band? That means the problem of non-specific or problem of non-target amplification. That is also a great problem. Okay, that's why people has devised different. You will find if you go, if you Google it, you will find lots of PCR primer making tools, online tools. Okay, or offline tools are also there. There are many programs and softwares, or you can make your own also. No problem. But to my point of view, to my experience, I can tell you, primer blast of NCBI, this is the best platform of making the PCR primers. Why? That I am going to discuss now. So this has already been discussed. Okay, that means actually what I want to say, PCR is a game of controlling temperature something. Okay, then okay, now the turning point. That means whenever you design a primer, an ideal primer should have the following properties. Okay, and what are those properties? The properties, that means to be an ideal primer, that means whatever problems that I have discussed, that is not going to happen. Your primers, the ideal length is almost 18 to 24 bases. 18 to 24 bases. Okay. Then what is the second? The second thing is 40 to 60% of GC content. You cannot increase the GC content because after lots of research, people has devised this. Okay, that means the ideal, if the length of primer is like this 18 to 24, it is ideal. You cannot make a PCR primer of 200 base pair long. No, it will take very a lengthier time to be annealed. So PCR reaction is not going to happen. Or if you make very short primer, suppose of 3 base pair long, so you never can make a very specific primer. That means a annealing of at a very specific site, because if it is very short in length, suppose 2 base pair, 3 base pair, or 4 base pair, probability of getting its complementarity, it will be very high. That means instead of getting 1 band, you will get 500 bands. Okay, so you have to maintain an optimum situation. Okay, so that's why these parameters have been devised. After doing PCR and lots of research, people have made. Okay, in an ideal primer, these properties must be there. That means if the first 1 or 2 base of your primer, if it start and end with maybe G and C, you will get better result. The annealing temperature, it should range between 50 to 60. Suppose your primer is so big, the annealing temperature is more than 72 degrees centigrade. That means the temperature of polymerization. What will happen? Your polymerase will be activated, so actually you will get no results. So, and then the primer pair should have a synchronization with its melting temperature with each other. That I was discussing. Okay, that means the forward and reverse primers, their annealing temperature should not vary much. I would not say 5 degrees. It is better if it is within 2 degree to 3 degrees centigrade. Okay. And the primer pairs should not have a complementary regions. That means suppose in forward primer, there is a region of suppose GC, GC, GC. And in the reverse primer, there is CG, CG, CG. So what will happen? Due to the laws of kinetics, instead of binding to the template strand, your primer will bind with themselves. Okay, that means primer dimer will be formed. Suppose in the starting region of your primer there is C, 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 and at the end there is G, G, G. So what will happen? There will be a self-complementarity, and your primer will form a loop-like structures. Reaction is not going to happen. So now think, why designing a primer, how many parameters you have to keep in your mind? So many times this is impossible to abide by all these laws, and you are making a primer manually. Okay. So actually, you have to make 2 primers, that is forward and reverse, with similar melting temperature. You have to make a balanced GC content, because too much GC content actually it will promote the self-complementarity. Okay, and its annealing temperature will also be increased if there is GC content is very, very high. If you can remember the formula that I have told you, that is 2 into A plus T plus 4 into G plus. That means implication of GC content is more as it is multiplied by 4. Okay, and you have to avoid this self-complementarity. You have to avoid this hairpin loop, and most importantly, the target specificity that I was discussing before. You have the genome length is very, very high. So there are many similar regions. Many times the similar genes are present in different locations of the genome. So it is obvious there will be sequence similarity. So you are thinking that you are going to get a single band, but actually after gel electrophoresis you have got 13 bands. Okay, so target specificity is very, very important. That you can never do by manual, because you cannot scan the whole genome sequence, and you cannot just find, okay, I need to find that what are the hotspots where non-specific targets are there. Okay, so though I am not discussing about RT-PCR today, if you are designing RT-PCR primers, then you have to avoid, okay, that your intron-exon junctions, because for RT-PCR you need to amplify the mRNA only. So that intron-exon spans, avoiding those things is also important. That I will discuss in another lecture. Okay, so in my opinion, primer blast is the best option. Okay, so what is actually this primer blast of NCBI? It is actually primer 3. You can get the primer 3 also. Okay, primer 3 is also a tool for designing PCR primer. It is also very popular. So NCBI actually they are using primer 3 program, but they have additionally included the blast into that primer 3 algorithm. Okay, so what additional benefit you get? That means that you can check the target specificity. If you do the blasting of your primers, you will be able to know apart from your target sequence, at how many points actually your primer is going to bind. That you can check before doing the PCR. Sometimes this primer blasting is known as ePCR. ePCR, okay? And most importantly, the integration of BLAST allows the screening of candidate primers against unwanted sequences of the organism's DNA. That is very important. And if you use this primer BLAST, you will get a graphical representation also. Okay, a visual representation also about the binding points. I will just show you practically. So this is, you can take this URL. Here you will find the primer BLAST tool where you can design the primers. Now I will show how can you design a primer using primer BLAST. Okay, so I'm just leaving this screen and I'm just going online. I will just show you practically how you can design the PCR primers using primer BLAST. Okay. Are you able to view my screen?
Yes, sir.
Okay. Okay. So this is the site of primer BLAST. Okay. I think this is very common today, this searching of nucleotides or searching of genes. So first I will show one primer designing from accession, which is already available in the NCBI database. Then I will show if you have any unknown sequence and there you want to make your or design your primers from both of that. Okay. Suppose this is your target. I have already searched this gene. Suppose this is your target gene. Okay, one Oryza sativa detoxification 42. This is the loci number. Okay, whatever maybe. Suppose this is your target. You can take any sequence of your interest. I have just taken this for example. So by FASTA format, or you will get here also the sequence. So in NCBI, you will get the pick primers options already here. Suppose you have downloaded one sequence and you want to amplify this this sequence for your own germplasm. Okay, so you will get the pick primers option here. So you just click here, the pick primers. Okay. So you see the accession number has already been put here in the primer BLAST tool. You see, this is the primer BLAST tool that I have already shown you the URL or website. This is, you have already given the PCR template, it has already taken. Now, there are some many parameters you can see here. What are those? Okay. So, if you have any range, suppose this is a whole gene. For many researchers or molecular biologists, suppose there is no need of doing the or making the primers from the whole genome. Maybe your target is different. So here you can define that means from where you want to generate the forward primer and where you want to stop. Okay, so you can put here, suppose you want to get the primers from 300 base pair to up to 800 base pair, from 300 to suppose you are giving one range 22 or 23 base pair like this, you can change it. But I will say, okay, make a this as default. Whatever. Put nothing here. Okay. You see what is happening. Or you can set your own primer also. Suppose you have manually designed your primers and you want to check whether everything is correct or not. So there also you can put your own design primer also and you can check. Okay. Another, that is PCR product size. By default, it is given minimum 70 base pair amplification, maximum 1000. You can change it as per your need. Suppose I need 252, suppose 700. Okay, and in return, how many primers you want to get? So by default, primer blast will give you 10 results. That means they will suggest you 10 PCR primers. You can increase it, you can decrease it. But here I have kept it as 10, okay, by default. And the primer melting temperature range, it is very important. Minimum, you have to set it. Minimum 57 degrees centigrade, optimum 60 and maximum 63. You can change it. Here, that how much difference between forward and reverse primer you are allowing. Here, by default, it is 3. You can make it 5, you can make it 2, you can make it 1. Okay. Then exon intron selection, no need to do anything, because if you are going for RT-PCR, then you will need this. So whatever is by default is there, you keep it. Then the organism, they have by default has taken the organism because it was rice. So the primer blasting has to be done in rice, in the similar genome. Suppose you are, if your organism is the gene you have taken from human, you cannot take the genome of a mouse for scanning. So whatever the gene from whichever organism you have taken, you have to set that organism. Okay, or it has already taken the organism name, or you can write here also. Oryza sativa. See, lots of options has come. Oryza sativa, japonica, aromatic, japonica. So whatever what is your target organism, you just select that. Suppose your group of organism is Oryza sativa aromatic, you can give aromatic. If it is japonica, okay. So whatever is available, you can. Just I have given this, okay, that Oryza sativa, japonica, cultivar. It will just scan this genome only, that whether any non-specific binding will be there or not. Okay, or you can add your organisms also. Okay, otherwise everything you keep as such or as default. Then you just click here the get primers. Then you have to wait for a while. Yes, this screen will come. You shouldn't do anything here. Just wait. Automatically the result will come out. And if there is so much of non-specific binding, they will tell you that also. That means you shouldn't go for this, that they are unable, because chances of non-specific binding is very, 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 very high. Okay, then you have to move. Okay, so how much time it is taking, everything you will find. So do not do anything in that screen. Actually, lots of time is taken for that blasting. Okay, actually it is scanning the whole genome That's why the time is required. Because genome is very, 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 very huge. And I have already given for the organism, that means whatever the information is available here for Japonica, it will scan everything. So primer blast is doing its job. Yes, it has come. Let the graphical representation come. Yes. So it is very beautiful. So this is the original template, this black color. Okay, you can see this black color. This is the original. That means the genome. And here specifically you have targeted this gene, this red color. Okay, you want to amplify, you are going to amplify this gene from this whole genome. This particular gene I want to amplify, and this is the location ID, the gene ID. Okay, so if you use the primer pair 1, that it has already, the sequences have been given here. If you use this primer pair 1, this much amplification will go on only. You can see this is the left border, this is the right border. That means out of this whole gene, only you will get this much of amplification. Okay, but your target was from here to there. But look, practically it is not possible. If you want to get very, 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 very specific, that means very target specific amplification. Okay, I had given, okay, you make the primers from here to there, but it has, if you use the primer pair 1, it will be only this portion will be amplified. Okay, then primer pair 2. If you use this primer pair 2, okay, this is amplification. You will be able to amplify only this portion. That means it is going beyond your gene, okay, your target gene. Okay. If you use primer pair 3, then this portion will be amplified. Okay, I think now it is clear. That means as it has given you 10 pairs of 10 primer pairs, I think now it is clear to you that if you use using which primer pair, you can amplify which region. So if you ask me, I will prefer either primer pair 3 or primer pair 4, because they are the almost the longest region can be amplified very specifically within my query sequence. Okay. So what is there in primer pair 3 or 4? Okay, have a look. Primer pair 3. Okay. See the length, it has the forward primer, okay, 20 base, here also 20, no problem. And see, their melting temperature is so close. For one is 60.18 and for another is 59.97. So it's very, very ideal, very good. And GC content is somewhat high. It should not be more than 50%, but there is a problem. GC content is 55%, but I think it will work. And cell complementarity for this primer, there are 5 bases where self-complementary can happen. Okay, and this is self 3' complementarity for one, and for this it will be 0. So I can use either primer pair 3 or I can use primer pair 4. So have a look at the primer pair 4. That is also almost similar with this. So I think there will be no problem if I use either of primer 3 or primer 4. Okay, so I think now you will be able to take your decision for making any primer, because out of all these primers, actually which primer pair I am going to use. Okay, but yes, if I click here, that means specificity of the primers. What they have written, primer pairs are specific to input template as no other targets were found in the selected database. That means Japonica group. That means there will be no problem. Okay. So again, I am doing this where keeping everything default. Okay, I'm not going to change the organism or anything, whatever, because they have, it has, what is the organism ID? They have already, by default, it has taken this. See what happens. So you have to wait. You have to spare this time, because this unique option of primer blasting is available here. Yes. Okay. So when it has chosen its more specific organism, you can see here the primer 5. It is the longest one. So if you ask me, I will choose primer pair 5. So what is there in the primer pair 5? length is same. Okay, temperature of melting is also same. GC content is fine. Okay, so I think you now question may arise that what is self-complementarity and again self-3' complementarity. Actually, self-complementarity, that means whether primer, dimer is whether hairpin loop or there is any chance of that means bonding with itself will happen or not. What is the chance for how many? This is a score for how many base pair it can happen or not. But 3' complementary, actually polymerization reaction happens at 3' end. Okay, addition is done at 3' end. So that's why additionally the 3' complementary is more important out of this self-complementary. That means it will actually, this score is showing you that whether any chance that the primer will bind to itself and also to other primers just at 3' end or not. If the 3' ends is filled by making any primer time or maybe here, that means everything is okay, but hairpin loop at just 3' end only, it will create another problem. So additional to self-complementarity, 3' self-complementarity, it is also another important, because if 3' end is problematic, there will be no chance of addition of DNTPs and actually no progression of amplification, DNA replication or polymerization will occur. Okay, and ultimately we will get no result. Okay, so as I have shown, if you take the DNA sequence or the gene sequence from the database itself. Now I am going to show you, suppose you are having some unknown sequence, you are having your own sequence, okay, that has not been deposited. There is no accession number, nothing. And that you want to amplify in your own germplasm or in your own samples. What you are going to do? Okay. So in that case, okay, then you have to go, just you Google primer blast. Okay, so there you will find this tool, primer blast. Only this window will open. And suppose this is your own sequence. Suppose this is my own sequence and I have to put it in FASTA format only. And I want to get the primers. Just you paste here and simply you click get primers. So here also you have to wait for the primers to come. I deliberately made one mistake here. Okay, knowingly I made a mistake here because sometimes it happens. Actually, the the sequence that I have used, that was taken from Oryza sativa, that means from rice. But machine doesn't know everything. That means it is a software. And here, as reference sequence, it has used the Homo sapiens. So, suppose if you make this mistake, if you just while doing this, if you just click get primers, every time you will get some results, but this result has no meaning. Okay, because I want to amplify one rice sequence, but suppose I have mistaken that I didn't specify the organism, so by default it will take Homo sapiens. And you see here also it is showing okay primers will, but actually if you order these primer pairs and you go for making reactions, you will not get these amplifications. Okay, so don't forget to add your organism here. That means which organism you are going to scan for your target specificity or non-specificity. So by default, there will be Homo sapiens, and some results will come every time if you just put and click the get primers. So if it is suppose E. coli sequence, you have to write here E. coli. Maybe it is unknown, it has not been reported yet, but the sequencing has been done in some organism. Okay, so you just put the name of that. I have used Oryza, so I will write here Oryza sativa. Oryza, but I may not specify everything. I just I can specify maybe Japonica, aromatic, template, tropical, aromatic, Japonica, many things. But here I have just given Oryza. So whatever Oryza will be there, it will scan everything. So then I am just clicking get primer. So never do that. Don't forget to fix your organism here while doing the PCR primer blast, because result will come, and by knowingly or unknowingly you can order the primers, your money will be just wastage. This specific feature you will find exclusively in this primer blast. That's why I prefer primer blast. Look what they have told. I have used some unknown sequences, but that PCR template is highly similar to the following sequence from search database. So to increase the chance of finding specific primers, please review the list below and select all sequences. That means whatever sequences I have given, it has found the similarity, 99.84% similarity with the sequence of this database. So they are asking, are you using this thing again? Because I am telling you unknown because to think it is unknown, but I have taken from this only. So they have just selected. Okay, that means, are you using this sequence? I am just selected this and submitted. If you have used complete unknown sequence, I have just taken as example. So if complete unknown sequence, then this message would not have come. But if you have copied from somewhere and you are pasting that, they will, by scanning the whole organism, whole genome, they will search everything and they will tell you. That means, are you going to amplify this sequence? So you have to spare this time. You have nothing to do. And if your internet is slow, little bit slower, then it will take much more longer time. Okay. Yes. Now look, for my sequence, so, they have advised me, okay, you can use these 10 primer pairs. So out of that 10 primers pair, if you ask me to choose, then I will choose either primer pair 1 or primer pair 10 or 7. Okay, so all are of very good, only except primer pair 2 and primer pair 8. They are very, very short. So I can use any of these 1, 3, 4, 5, 7, 9, 10, anything I can take. Okay. So just I have to check the self-complementarity and other parameters. Okay, whether there is any synchronization between the melting temperature or not. Self-complementarity score is very high for this. So I shouldn't go for this primer pair 1. Okay, suppose this primer pair 4. Okay, the TM is almost identical and these scores are also very, very less, 4 and 3. So it is better to go for primer pair 4. Okay. So I think now you will be able to make your own primers for your own research. So the today's discussion is over. So if anyone have any question or any doubt, so I'm ready to answer it. Okay, so thank you all for keeping patience and listening to me. So thank you everyone.
Thank you so much for this very informative session. If anyone has any question, you can ask. I think no one has any questions, sir.
Okay.
Just to inform you all that this complete session will be available on our Research Circle YouTube channel. So if you want to watch it again or want to share it to somebody, please do Research Circle YouTube channel. Thanks, and last but not the least, thank you, Dr. Sandeep Debinath, for taking out the time for the session. With your permission, I would like to end today's session.
Okay. Thank you. Thank you. Thank you, everyone.
Thank you so much. Thank you, sir.